Hi everyone, I hope you're doing well. I was trying to find you a good video on this, but they were quite long, and I know that you guys have been taking a lot of notes lately, so I just wanted to be able to talk to you myself and to share with you some important things from this last unit. So you're going to work side by side with this slideshow. You might have to open it up in Google Slides, but it came from PowerPoint. And you're also going to side by side use our very last page of our cell biology packet here. I've opened it up in Kami so that I can write in it and help you through this last slide here. I might also give you guys some time to work on some parts on your own as well. So right here, very first thing, what is mitosis? I think by now you all know what mitosis is, but it's that dividing of the cells. And why is it important? Why do we care about mitosis? Why is it important? So dividing of the cells, creating two daughter cells that are identical, right? So mitosis is creating those two daughter cells that are identical. Why is it important? Well, without mitosis, what would happen to us without mitosis? We need this to grow new cells or damaged cells. All right. So there's this whole process that's happening within the cell when it's undergoing mitosis, where it's setting itself up to be able to divide to create those two identical cells. And we look at this whole process between the cell cycle and interphase. So during a cell's lifetime, it grows into many, many cells. Now, certain cells actually do not go through mitosis. Think about maybe a brain cell. So I have a friend, a really sad story, that she was 40 years old. It was like right around her 40th birthday, actually, and she had a stroke. And she had certain cells in her brain that had died, and they do not ever go through mitosis. So if you look here at our cell cycle, you'll notice that mitosis in this pinkish orangish color is not as big as the rest of the time that the cell is in what we call interphase. Interphase is this orange part here where the cell is going through many different phases, different stages. During that time, maybe your cell is going through homeostasis and regulating what's going in and out of the cell through osmosis, regulating if water is going in or out, or diffusion. Maybe it's getting ready, preparing for mitosis. Maybe it's making proteins and the DNA is replicating. So all of these things are happening to prepare itself. It's an active period where the cell is growing. It doesn't necessarily always in mitosis. So those of you, my seniors that did our mitosis lab, we didn't see very many cells going through mitosis. We saw a lot in interphase. We saw that one nucleus that was in. So interphase is divided up into three stages: G1, S, and G2. So you can fill this in on your chart. You can pause me and fill it in as I'm talking. So during this cell cycle, G1 is kind of that period of growth and increase the organelles, right? If we're going to divide, we need to be making more of those organelles that are really important. So that's this period of growth that the cell is going through. S phase, or synthesis of DNA, is this replication process where DNA is being made and replicated so that it can actually be passed on into two daughter cells. And then G2 is where it's actually starting to prepare for mitosis. Maybe those chromatids start to come together. So we're looking at our chromatids right here. These are chromatids. And when it goes through this process of mitosis, when they line up in a process called metaphase, these chromatids start to come together. And this is what can actually start to prepare for this splitting. Cytokinesis is that final stage. You need to remember that it's producing two identical daughter cells. All right, so as of right now, you should have your page. I'm trying to find the spot where I want you, and it's not showing me exactly what I want. Here it is. Okay, so you should have what is interphase. You should have the 3 parts of interphase. Right? So I'm going to quickly go through these different stages of mitosis, but if you could add pictures in there and spend some time filling that in, that would be wonderful. So let me jump over to mitosis for you. So when you were doing those different stations, you worked through this. I'm sure you worked through this before. But once those cells start to divide during mitosis, they go through a few different stages to get there. So the stages are prophase, metaphase, anaphase, telophase, and each one something different is happening within the cell to create those two daughter cells that are exactly the same. So after your DNA is replicated during your S phase, this is a whole process that goes into place where the DNA actually splits and pieces of the DNA start to get replicated in order to make new strands for those daughter cells. So 2 copies of the chromosome is eventually what's going to happen here. And at the beginning of mitosis, these chromatids, okay, so you have your chromosome, in the center here is your centromere. And when it's replicated, we call those 2 exact copies of each other sister chromatids. They are exact copies of different chromosomes. You have 23 different chromosomes in your body, so all 23 would need to get copied with their sister chromatid. So prophase is that first stage that you have, and what starts to happen is your chromosomes begin to become visible, right? So they're all coiled in these long, huge strands, and they start to kind of supercoil up so that you can actually start to see them line up, and we did see some of that in our onion cells. So the centrioles will start to move to opposite sides of the cell and begin to kind of pull at these chromosomes. If you look here, the centrioles start to have these spindle fibers that come out and start to kind of organize those chromosomes in the middle. So we can see here that our DNA is all coiled up in our nucleus with these, but in here is where we start to see our mitosis happening. And you can say which of the following plant cells is in prophase. Which of the following plant cells is in prophase? Well, I haven't met in the middle yet, so I would probably say this one up here. Our next phase is metaphase, and that's when they'll start to meet in the middle. That's after your nuclear membrane is broken down, your spindle fibers fully attach, develop and attach to the centromeres, and they start to align up those sister chromatids along the center of your cell. Metaphase, meet in the middle. Okay, so we need something meeting in the middle, probably something similar to this little guy over here. After they've lined up, they need to then split back apart. Remember, the whole point of making the sister chromatid was to then split it back apart and make 2 identical cells. And we do that in anaphase. Away. They'll pull apart opposite poles of the cell, and now we have our chromosome again from our chromatid. Probably this one over here. I would call this metaphase, anaphase pulling apart. Maybe that prophase. Let me look again. Prophase. I would say probably then like this one. They're starting to unwind and starting the process. All right. Telophase is that final stage. Chromosomes are now arriving at the poles. They are getting ready for cytokinesis, where our cell finally will split. Ooh, beautiful telophase right here. And I think this is what we had seen in Graham and Ben and Rocco's slides, some telophase happening. All right, so you have this whole cell cycle happening. All of mitosis is in this part here, and it goes through and shows you each step, starting and all the way through to cytokinesis. All right, looking at our final couple questions here. Cytokinesis is that final part right after telophase. They're not technically calling it part of mitosis, so it's happening right after telophase, which splits and divides the cell into two, saying that it's a little more easier or simpler of a process for your animal cells versus your plant, and you guys know why. You have that cell wall you have to deal with, right? Okay, so as of right now, you should have filled in different stages here. What are the different phases of parts or phases of interphase, and now cytokinesis. So we're going to jump into this mitotic index and how it's calculated, which is something that we'll be doing for our next lab. So the mitotic index is a pretty simple equation that we can use to kind of say how many cells are going through mitosis versus the rest of the cells in that area of our microscope. So we're looking at a sample from what we call a micrograph. This is a picture of our field of view in the microscope, and this was something that I wanted you guys to be able to do from our last lab, but maybe you didn't find enough. So looking at this, the mitotic index of a 27 sample is the proportion of visible cells that undergo mitosis. So what does that mean? It's the cells in prophase, metaphase, anaphase, telophase divided by the total number of cells, and that gives you your mitotic index. So if you're looking at this, you could calculate out how many cells you can see in your field of view, and then in prophase, metaphase, anaphase, telophase, and then divide it by the total number. So if you were to look at this picture here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 34, 35 total. Now you have to go and calculate out how many are in prophase, anaphase, metaphase, anaphase, telophase, and then divide by the total amount. And same here, you can do it there too. So it's a really important tool that we use. Think about why. Why would it be important to figure out how many cells are going through mitosis at a time in comparison to the rest? And the reason would be cancer. So if we're looking at an area of someone's body that is going through cancer, you're going to see those cells in mitosis probably a lot more. They're replicating, causing possibly a tumor. And that is why we look at this, the number of cells that are dividing in comparison to the rest in interphase. So when a tumor is forming, our cells have kind of gone out of control. And it's really interesting because our body has certain procedures in place where they actually can stop this from happening. There's like cell suicide, where cells go through a process called apoptosis, where the cell will kind of just burst if it's not the right type of cell that's supposed to be there. There are also different things that will stop the growth of these cancer cells from dividing. And then sometimes we don't have that control, and they start to divide uncontrollably. And this is where you'll start to see a mutation happening, or you'll start to see that tumor. And this is the part that